All right, welcome to part 2. So as we left off, we talked about how, and you need to write this formula down, in a aerobic respiration, the reaction that takes place, what's going to happen is we're going to take glucose. Okay, so we're going to take some glucose. And aerobic, that key word, once again, means with oxygen. So with oxygen, and along with some ADP and some inorganic phosphates that are laying around, we're going to use some enzymes. Okay, remember, their proteins are reusable. We're going to use some enzymes, and those enzymes, okay, are going to help speed up the reaction. Without it, the reaction can't take place. The end result of that, our end product, we're going to get carbon dioxide. That's what we're going to breathe out. Plus, we're going to get some water and 38 ATP. So I'll say that again. Make sure you have this. You take glucose plus oxygen plus ADP plus PI. You need these enzymes up here. And you're going to get carbon dioxide and water. So we'll break down glucose to carbon dioxide and water, produce ATP. Net 38. Other key component of that, glucose, its formula, C6H12O6. Not to freak you out, if you go back to a little chemistry, and you remember you got to balance things out. So we got some uneven numbers. Well, we can fix that simply by adding a 6 right there to make 6 oxygen. 6 right there, that'll get 6 CO2. 6 max it up there. 6 right there. Now it's all balanced. And we put 38 and 38. So there's the formula. I'll repeat this. Glucose, which is C6H12O6, plus 6 oxygen, plus 38 ADP, plus 38 PI, needing enzymes, and you're going to get 6 carbon dioxides, 6 waters, with a total gain, okay, not net, but a gain of 38 ATP. And that's only when you do it with oxygen. The very first steps of this process, when we do this process, it requires ATP to get it going. We call that energy to get a reaction going the energy of, and I think you've heard of this, activation. And this energy of activation is what gets the thing going. For this particular reaction, we need 2 ATP. Okay? So right off the bat, before you get the party started, boom, you just spent 2 ATP. So it's like an investment. It's like if I told you, I can get you $38, but today you got to give me $2. Tomorrow I'll get you 38. It's still a good deal, but at the end of the day, how much money did you make? Well, if you paid 2 to start, I gave you 38 at the end, overall our net gain would be 38 minus 2, a total of 36 ATP. That's the number we need to remember. We need to remember that it's 38 is the total number for 38 is our total, but our net's the key, okay? Just like how much money did you actually make? Well, it's 36 ATP. That's how much you actually walk away with. You gain 36 ATP. So the maximum net ATP that can be produced is 36. I want you to remember that just like anything else. If I say aerobic respiration, boom, it's instant 36 ATP. This is going to be on your test. This will be on your semester exam. This is going to be on your final exam. Aerobic respiration nets 36 ATP. Now what's interesting with that is that, you know, whether that seems like a lot, 39% of the energy of that original glucose molecule, okay, is captured in that 36 ATP. Only 39%. The rest of it is lost. How's it lost? It's lost in the form of unusable energy, form of heat, or it remains in the end products of that carbon dioxide and water. Water! Okay? The purpose of this process is to slowly release the energy in glucose bit by bit into ATP. Here you can see the reaction. We've talked about it's an ATP synthesis reaction. In this case up here, when we go from ATP to ADP, we're using that energy and the breaking of that last bond. Remember, by breaking this last bond here, we're going to use that to do all these things. We are then going to take, in this case, glucose, break glucose down into carbon dioxide and water, and in the process, we're going to produce how much ATP? Net, if it costs us 2, make 8, the net is 36. Same thing here. So just another diagram showing that reaction. Okay. The reaction of aerobic respiration has 4 parts, and we're going to look at these today. There's 4 parts to this. What I want you to do right now is somewhere on your piece of paper, I want you to make a box, rather large box. You're going to divide it into 4s, just like this. And you're going to put glycolysis in one box. You're going to put next to it pyruvic acid in the top of that one. You're going to make citric acid in the top there, electron transport there. And what I'm going to do for you, by the time we leave, you're going to have a summary of each of those reactions. Okay, that's what's going to be really important. If you asked me what's a key, glycolysis, this whole process, it can be rather complicated. I'm not going to lie. But if we just remember to break it down into its key points, that's what's going to make it a lot easier. Okay. So, aerobic respiration. The chemical potential energy that we have in glucose is going to be slowly released and transformed in ATP using these 4 types of reactions. And once again, at the end of these 4, when we get done with all 4 of those, our net gain, please tell me, fill it in, 36 ATP. That's what we're getting. Okay? The breakdown of glucose into ATP, because the energy of ATP is more convenient for the cell use, we can send to other places. It's the cache of the cell. Okay, it's the key point to it. The first one that we're going to look at on the next slide is actually glycolysis. So glycolysis. If I had my box here, what I would want to do is go over what's kind of happening in this reaction. The first thing I would put in that box is its location. Glycolysis happens in the cytosol or the cytoplasm of eukaryotes and prokaryotes. So it's happening throughout the cell. Enzymes are needed for it, but it's going to happen anywhere throughout the cell. No restrictions on that. To get glycolysis going, whether in your bacteria or us, it requires 2 ATP. It gets the reaction started, it gets it going. The overall gain out of glycolysis, when it's all said and done, and there's a bunch of steps. We're not going to go through all 10 steps. Just realize it's very complicated. There's a lot of steps to it. But at the end of it, you're going to get 4 ATP. Okay, now, once again, if we started with 2, it costs us 2, okay, ATP. We gain 4. Our net gain, right, is a plus 2. Glycolysis, we get 2 ATP out of it. This step of the reaction, glycolysis, is anaerobic. Once again, an mean not, no oxygen needed during this step. Happening in the cytoplasm. And the original glucose, we took 1 glucose in, it's going to be broken down into 2 pyruvic acid molecules. Along with the ATP, we're also going to get 2 NADHs, which are just electron carriers. So therefore, on my little square, I have glycolysis. I just put happens in the cytoplasm of all organism. Is anaerobic, no oxygen needed. And the end result of this, we get the 2 NADHs, a net of 2 ATP, and 2 pyruvic acid. That's it. Now there's a lot of steps to it. I'm going to show you the steps in the video. In a video, we're not going to have to know them. Just realize there's a lot of steps to it. The next step is called oxidation of pyruvic acid or pyruvic acid oxidation. It's the same thing. Okay, let's go over what this is. I'd move over to the next box. Here's my pyruvic acid box. Let's see what we have in here. The first thing, there's no ATP cost, okay? There's no ATP gain. So what I did is the first thing that you have to know is it happens. Where it happens. It happens in the mitochondria of eukaryotes. And it's a process of changing the structure of those pyruvic acids that we had from glycolysis. Okay? These things right here. But notice, that's it. It happens in the mitochondria. It's aerobic, meaning oxygen. But notice, I said nothing about ATP. Didn't cost us anything. Didn't get anything out of it. I said nothing about NADH. So on mine, I put on there nothing, zero. None of that happened. All it does, it happens in the mitochondria of eukaryotes, and it changes that. Pretty simple. The next stage is called the citric acid cycle, also named Krebs after the person who discovered it. In my Krebs box, first thing I want this to say, it's the same as last time. So its location, this time, just like the oxidation pyruvic acid, happens in mitochondria. So it happens in the mitochondria of eukaryotes, okay? It's aerobic, meaning it needs oxygen, and it is a cycle. And a cycle obviously means that what we start with, we're going to do different steps to it as we go along. And at the end of the day, what we start with is what we end with. It's a cycle. Okay? So at the end of the day, we end with the same product. What we get out of this, the net gain, the important stuff you need to know about. I'll put that on here now. So the net gain of the citric acid cycle is 2 ATP. We get 2 ATP, we get 6 NADH and 6 H plus and 2 FADH. Now, these things right here, may not have heard of them, they're electron carriers. Basically what we're going to do is we're holding them for a little bit. Eventually at a certain stage that we're going to talk about next, we're going to convert those to ATP, but not right now. Now they're just in this form. Okay, citric acid cycle happens in mitochondria. It's a cycle, so what you start with, what you end with, and at the end of it, we have our products. The last stage of this process is the electron transport chain. In this box, first thing, it's our payoff. This is our payoff. Because so far we said, how much ATP should we get out of this? Well, we should get 36 ATP, right? Net. It's a net ATP. But so far in glycolysis, we only got 2. In oxidation pyruvic acid, we only got 0. And last citric acid cycle, we got 2 for a net of 4. So obviously, what does that mean? Where's the rest come from? Ding, 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 ding. Good job, Z-Tech. Electron transport chain. That's where the energy is going to come from. So let's talk about what happens here. So it happens in the mitochondria again. Last 3 all have. And it's going to take the energy from NADH. Okay, we had, remember, if you'll take a look at that NADH, we produced 2 NADHs in glycolysis. We produced 6 NADHs in citric acid cycle. We produced 6 of these in the citric acid cycle. And we produced a total of 2 of those in the citric acid cycle. You take all of the energy of those, and in this process, in the ETC as it's known, the electron transport chain, we're going to convert the energy of those into ATP, producing 32 ATP. So as a summary, glycolysis, we got 2 net, cost us 4. And oxidation of pyruvic acid, we got 0. In citric acid, we got 2. And ETC, that's where our 32 is. 32 plus 2 plus 2 is our total of 36 ATP, which you should always know when you do it aerobically. So as a review, here's what we have. We got glucose, that's this on the left, plus oxygen, because it's CO2 and water. And here's the actual process. when you take a look at this, glucose, this is the different steps. I like this diagram. It shows where it happens, which is important for us to see. Glycolysis happens in bacteria, happens in the cytoplasm. Okay. Formation of acetyl coenzyme A, also known as oxidation pyruvic acid. That's what we're going to call it. Citric acid cycle and the electron transport chain.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into two molecules of pyruvate, energizes two electron carrier molecules, and produces two molecules of ATP. The next two stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create two more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
In muscle cells, aerobic respiration takes place, but after an extended amount of time, what ends up happening is it stops performing it. And it does a different type of reaction called anaerobic, without oxygen. So you start it that way, you run out, and you start doing anaerobic respiration. And during anaerobic respiration, because we don't have oxygen, we can't do steps 2, 3, and 4 of the process. We can't oxidate pyruvic acid, we can't do the citric acid, we can't do electron transport chain. So what does that mean? Well, look how many ATP are found in those last 3 stages. 2 from citric acid, 32 from electron transport. Those 34 ATP that are done in the last 2 stages are lost. And when we do anaerobic, we only get a net gain of 2 ATP. It's a huge difference than what we started out with. Some bacteria will carry out all their life processes without ever using oxygen. They're anaerobes. Okay? And the 2 different types of aerobic respiration are lactic acid fermentation, mainly done in us, and alcohol fermentation. Your homework for next time is the following. Your homework, read 6.1, define lactic acid fermentation and alcohol fermentation. Do that right here in the notes that you've been taking through this video. Write down the formulas for each. Answer questions 1 through 5 on page 151. Boom goes the dynamite. That is it. I'm out. So are you. God bless America.